Good afternoon, dear students scattered all over the Gujarat of the subject microbiology. Myself, Parul Bhatt is here with my colleague Ami Purohit from Microbiology Department, J&J Science College, Nadiad. Friends, we are today here for the lecture in BISEC to talk on the subject of role of microorganisms in biogeochemical cycle. Friends, what is biogeochemical cycle? It is the cycling and recycling of elements or chemicals through number of assimilation and decomposition reactions involving various forms of life of different ecosystems in the biosphere. The definition is too long. To make it simplified, it is just the conversion of elements from one form to another form with the help of microorganisms. Since the question may arise in our mind, since when all this cycling and recycling initiated? And what was the atmosphere on Earth at that time? How the atmosphere changed? How the life originated and how it evolved? To have the answers to all these questions, let us see some glimpse of origin of life on Earth. Friends, we are talking about atmosphere of Earth before 4.5 billion years ago. At that time, Earth was not having today's environment or today's atmosphere. The atmosphere was very primitive. There was no oxygen, there was no ozone, there was no life, and Earth was very cold. UV radiation from the sun was keeping the rocky surface of the Earth completely sterile. Earth was having molecules like carbon dioxide, sulfate, ammonia, iron, etc. Now, let us see where was the possibility of life and how the material to give rise life assembled. Then the answer to this is there was certainly no possibility of life on rocky surface of Earth, but at that time ocean was already formed due to heavy rainfall on Earth, and in these water bodies there was dissolved form of sulfate, ammonia, iron, carbon dioxide and many more chemicals in water. This water was having ice caps. So the role of ice cap was very good in the favor of life. Ice caps prevented the direct radiation of UV rays in this chemical containing water. As a result, chances were there for life formation. Initially, it could have produced, we are not certain, but it could have produced primitive organic chemicals like formaldehyde, HCN, ammonia. This may have somehow formed amino acids like, for example, lysine. Now, any chemicals, if they want to react, they require energy. Here the molecules got energy from natural sources because at that time there was lot amount of lightning on the Earth. There was continuous eruption of volcanoes. There were hot springs and there were meteorite impacts on the Earth. All these provided needful energy to catalyze molecule formation needed for life. Catalytic molecule could have generated somehow. It is believed that two aldehyde phosphates could have reacted to produce sugar phosphate, and from that arised first genetic material on earth, that is RNA, full form ribonucleic acid. Still the earth was very hot with continuous volcanoes eruption, number of hot springs, then lightning was providing energy, and all these chemicals which we just now discussed, they led to one more chemical formation was formed. It added to this, that is very essential organic molecule for life, that is acetic acid, needed for catabolic reaction as well as synthetic reaction, that is anabolic reaction. Somehow in this soup, this is known as soup, it is very similar to the content which is present in the cell, which is protoplasm. This soup, in a fashion, it is known as primordial soup. This primordial soup contained all these now vital elements, including acetic acid and RNA, some catalytic elements for the life. Hydrocarbon molecules possibly were generated, which were polymers, and somehow it formed microspheres. What are microspheres? It is shown in the slide, yellow colored balls. It acts just like cells, and these microspheres, it enclosed this primordial soup, and this could be probably first primitive life on earth. However, first life on earth is believed to be chemosynthetic microorganisms. Probably they were sulfate reducers and ion oxidizers, because both of these molecules were present in the environment or the atmosphere. It is also believed that cyanobacteria could be the common ancestor of life. Then, how this oxygen generated? Today it is, we know the content of oxygen, it is nearly 21% in the atmosphere. How it is generated? At that time, photolysis, that is due to UV rays from sun, it caused photolysis of water and carbon dioxide. In the slide it is saying that CO is written, please read it as CO2. Photolysis of water and carbon dioxide generated oxygen. This oxygen was initially absorbed by iron. Iron was present in the earth in the form of Fe+2. By absorbing this oxygen, it got converted to FeO, iron oxide, and the figure which is seen in the slide, it is of banded iron formation, red in color. Still evidences are there on the earth. Then when this reaction was on the saturation, means when complete iron was in the form of iron oxide, newly emerging oxygen was now gathering in the atmosphere. This oxygen, once again it was radiated by UV rays, and from oxygen it formed O3, that is ozone. And we know we have heard about ozone since long back. Here in the slide you can see ozone layer in shaded band between Earth's surface and sun. What is the role of ozone? It filters UV rays, that also we know. So at that time this ozone layer was now creating. What is the result? Result is new atmosphere, that is, it is well known as secondary atmosphere on Earth. So the atmosphere in which we live is secondary atmosphere. Up till now it was primitive atmosphere. By the generation of ozone, it has formed secondary atmosphere. Conditions were becoming favorable for photosynthetic organisms evolution. It is believed that organisms like bacteria and algae capable of photosynthesis were evolved, and they generated still more amount of oxygen. Atmosphere was now day by day enriching in oxygen content. Then life forms like fungi and algae evolved. Symbiotically they colonized rocks, and these lichens are known as responsible factor for soil formation. So now try to link the things. Atmosphere is favorable, oxygen is now gathering, and soil is forming. Conditions is changed. And before 400 million years ago, lowland plants were generated. They started evolving. This was followed by upland plants, means high rise, huge plants developed, resulting both of these resulted in huge organic matter on the earth's surface. Previously, organic matter was at all absent on the earth in primary atmosphere. Now there was accumulation of organic matter. It was before 100 million years ago. This was followed by evolution of angiosperms, that is flowering plants evolution. It was approximately 1 million years ago Homo sapiens evolved, and before 500,000 years ago man evolved. It was around 8000 BC. Till that, this primitive man, newly evolved man, was just gathering food material from nature or by hunting. But it was around 8000 BC when man started growing his food and for that he used to clear new land for better plants. It was between 3000 BC to 700 BC. BC means before Christ. Actually, agriculture practice started. It got refined, it got more refined, like that. And so today we know, we are very well aware of agricultural practices. We are living in the age of what we can say GMP, also genetically modified plants. We know we have fertilizers, biofertilizers, irrigation and all that. So life is evolved quite much. When we go through this journey of evolution, it seems clear, or it becomes clear, that solar energy created photosynthetic life, or it initiated photosynthetic life in primary earth atmosphere. In turn, the atmosphere changed and the condition became oxygenated on this earth, which is known as secondary atmosphere. So friends, when we recall the things, it becomes clear that it is the atmosphere that evolves the life, and it is the life that changes the atmosphere. It is both the way true. So jointly, we cannot differentiate any one of this. This is a joint journey, the origin of life on earth. And since then, the vital elements for life, which are carbon, nitrogen, hydrogen, oxygen, phosphorus, sulfur, have undergone a series of recycling processes through assimilation and decomposition, thereby creating a biosphere. So let us now have a systematic study of these recycling processes, which are very well known as carbon, nitrogen, hydrogen, oxygen, sulfur, phosphorus cycles. My colleague Ami Purohit will take you in this lesson.
Thank you. As my colleague told, biogeochemical cycle involves movement of the important elements of life in the different parts of ecosystem. That is movement of carbon, nitrogen, sulfur, iron and phosphorus among atmosphere, stratosphere, biosphere, hydrosphere and lithosphere. Let us see individual movement of every cycle in detail. Starting with the carbon cycle. As we all know, carbon is the first element required for every life. It is an important precursor for carbohydrate, fats, proteins and nucleic acid. Human body contains about 18% of the carbon, which is 100 times more than that present on the earth. Carbon cycle was initially discovered by Joseph Priestley and Anthony Lavoisier and was popularized by Humphrey Davy. First of all, let us see where is all the carbon stored in the planet. It is present in the form of carbon dioxide in atmosphere, in the form of soil organic matter, as living and dead organisms in biosphere, as fossils and rocks in lithosphere, and as dissolved carbon dioxide and in shells as calcium carbonate in hydrosphere. Carbon cycle is just a movement of atmospheric carbon dioxide to organic carbon and again release of organic carbon dioxide back in the atmosphere. That is a movement of inorganic carbon to organic carbon and again to inorganic carbon. It is carried out in two steps. Let us have a glimpse of this cycle first of all. The atmospheric carbon dioxide is fixed in plants and photosynthetic bacteria. In plants, it is fixed in the form of proteins, cellulose, hemicellulose. Plants are eaten up by animals and so it passes in the animals in the form of carbohydrates, proteins and fatty acids. When these plants and animals respire, carbon dioxide again enters the atmosphere. Moreover, when these plants and animals die out, the dead organic compound gets deposited in the soil. This organic compound in the soil is then further degraded by soil microorganisms, which releases carbon dioxide again back in the atmosphere. In spite of all this, burning of fossils and fuels also releases carbon dioxide back to the atmosphere. Let us see the cycle, individually steps of the cycle in detail. The first step is conversion of inorganic carbon dioxide to organic carbon. It has been seen that autotrophic group of bacteria, through reduction processes, uses carbon dioxide to form carbohydrates. Moreover, during photosynthesis, plants uses atmospheric carbon dioxide to prepare their food. Thus, the atmospheric carbon dioxide is used by plants and photosynthetic bacteria to prepare organic carbon. Moreover, heterotrophic microorganisms also utilize carbon dioxide. For example, pyruvic acid when combines with carbon dioxide forms oxaloacetic acid. Thus, the atmospheric carbon dioxide is now present in plants and animals in the form of cellulose, pectin, carbohydrates and so on. Our second step is degradation of organic compound. The most abundant organic compound present in the soil is in the form of cellulose. Cellulose is degraded to cellobiose by enzyme cellulase, cellobiose to glucose by enzyme beta-glucosidase, and glucose by glycolysis releases carbon dioxide back in the atmosphere. These reactions are oftenly carried out in a protocooperation method by Cellulomonas and Bacillus group of organisms. Thus, we can say that the above two reactions are reversible. One's reaction's products are the other's reactant. Similar pathways are also available for degradation of hemicellulose, lignin and pectin. Moreover, CO2 may originate from the decarboxylation of amino acids and fatty acids. In spite of the basic cycle, it has also been seen that the burning of fossils, fuels, and the respiration of plants and animals also releases carbon dioxide back in the atmosphere. So this was a general look where we have seen that the atmospheric carbon dioxide is fixed in the soil and again released back in the atmosphere. Coming to our next cycle is nitrogen cycle. Nitrogen was initially discovered by Daniel Rutherford, which was known as noxious air. It is an important building block of proteins and nucleic acids. Human body contains about 3% by weight of nitrogen. Nitrogen cycle. What is nitrogen cycle, or what does it mean? It is a sequence of changes from free atmospheric nitrogen to inorganic nitrogen to simple organic compounds to complex organic compounds and eventual release of this nitrogen back to the atmosphere. Nitrogen cycle thus comprises of 5 steps, that is 5 oxidation reduction steps. Probably this is the longest cycle as compared to the other elements on the earth. Let us have an overview of this cycle. Atmospheric nitrogen is fixed by symbiotic and non-symbiotic nitrogen fixing bacteria in the soil. This fixed nitrogen is converted in the form of organic nitrogen, that is in the form of proteins in plants. Plants, when they are eaten up by the animals, the nitrogen passes in the animals and gets deposited as a part of soil organic nitrogen. This organic nitrogen then undergoes degradation. The process of degradation releases ammonia. Ammonia further undergoes ammonification. After ammonification, from ammonia, nitrite is formed. Nitrite further forms nitrate, and nitrate reduces nitrogen gas back to the atmosphere, that is denitrification. In spite of this basic cycle, there are intermediate steps occurring in this cycle. For example, nitrate is oftenly used as plants for their food. Moreover, the same nitrate under anaerobic conditions is converted to ammonia, and ammonia also is used as a source of nitrogen for various plants. This was a general overlook of nitrogen cycle. Let us see all the 5 steps in detail. Coming to the first is nitrogen fixation. Nitrogen fixation means conversion of the atmospheric nitrogen to ammonia. It has been seen that about 100 to 200 million tons of nitrogen has been fixed through biological and non-biological processes, from which 2/3 is fixed by biological process only. Nitrogen fixation occurs in 3 ways: atmospheric fixation, industrial fixation, which is also known as Haber-Bosch process, and biological fixation. Atmospheric fixation. It has been seen that 5 to 8% of the fixation process occurs in this way. The excess lightning breaks nitrogen molecules apart, which combines with the oxygen molecule to form nitrogen oxides. Nitrogen oxides dissolve in rain to form nitrates, which are carried on to the ground along with the rainwater. Industrial fixation. At high temperature of about 600 degrees Celsius and great pressure, using a catalyst, atmospheric nitrogen and hydrogen are combined to form ammonia, which is used as fertilizer. And the third is biological fixation, which occurs by 2 types of bacteria, that is free living bacteria and symbiotic bacteria. Symbiotic bacteria fixes about 70% of the nitrogen, while free living bacteria fixes 30% of the nitrogen. Let us see the mechanism of biological nitrogen fixation. It was initially described by Herman Hellriegel and Herman Wilfarth. It has been seen in the equation that 2 moles of ammonia are formed from 1 mole of nitrogen at the cost of 16 ATP, 8 electrons and 8 protons. Prokaryotes can carry out this reaction as they have enzyme nitrogenase. Nitrogenase enzyme consists of 2 proteins: iron protein having a molecular weight of 60,000 daltons and molybdenum iron protein having a molecular weight of 2 lakh daltons. This is the mechanism of nitrogen fixation, the reaction which occurs during nitrogen fixation. As shown here, initially the oxidized Fe protein is reduced by electrons donated by ferredoxin. The reduced Fe protein then reduces molybdenum iron protein by taking up 4 ATP. Reduced molybdenum iron protein then uptakes 2 hydrogen atoms and 1 mole of nitrogen and converts it to diamide. By further similar 2 cycles, diamide is converted to hydrazine and hydrazine is converted to ammonia. Thus, 1 mole of nitrogen results in formation of 2 moles of ammonia. The electrons which are donated for these reactions may be donated during photosynthesis, fermentation or respiration depending on the type of the organisms involved in nitrogen fixation. It has been seen that rhizobium and pedirhizobium are the organisms which successfully fixes nitrogen symbiotically in leguminous plants because they have an enzyme nitrogenase. This nitrogenase enzyme consists of genes like NOT genes, exogenes, NIF genes and fix genes. The symbiotic nitrogen fixation in leguminous plants was discovered by scientist Bergering. Free-living organisms like Azotobacter have also been seen to fix about 30% of the atmospheric nitrogen in the soil. Azotobacter oftenly form cysts. They are microaerophilic in nature and hence possess a high respiratory rate. Because of this, they soak up the free oxygen which is present in the cells and helps the nitrogenase enzyme to fix nitrogen because nitrogenase enzyme requires low oxygen tension to fix atmospheric nitrogen. These are the examples of nitrogen fixing organisms. Rhizobium and pedirhizobium fixes symbiotically nitrogen in leguminous plants. Frankia with ulnus is an example of symbiotic nitrogen fixation in non-leguminous plants. Azolo with anabina is an example of symbiotic fixers in higher plants. While the free-living nitrogen fixers are Azotobacter, Clostridia, Azospirillum and Bergering. Now, what is the fate of the fixed nitrogen? As we know, nitrogen has been fixed in the form of ammonia in the soil. Ammonia is incorporated into amino acids and amino acids bind together to form protein. Thus, the atmospheric nitrogen is now present in the form of protein in plants. The plants are eaten up by animals, so it passes in the animals. When these plants and animals die out, their debris get deposited in the soil, which comprises mainly of the organic nitrogen compound. This organic nitrogen compound now has to be degraded. So, our second step of the nitrogen cycle is organic nitrogen degradation. It is oftenly known as proteolysis, that is, degradation of proteins to peptides and peptides to amino acids by enzyme proteinases and peptidases. Organisms like Clostridium, Proteus and Bacillus present in the soil can easily carry out this reaction. The protein has been degraded. Now what? The third step is ammonification. Ammonification is also known as deamination, that is removal of the amino group by enzyme deaminase. When ammonification occurs, ammonia is released out. Ammonia is a volatile substance. It leaves the soil. But if it is solubilized in the wet water with water molecules, it converts itself to ammonium ions. And these ammonium ions participate in the further nitrogen cycle. So, our fourth step is nitrification. Nitrification is an example of complete aerobic process which occurs in 2 steps. As shown here, in the first step, ammonia is converted to nitrite with the help of ammonia oxidizing bacteria like Nitrosomonas, while nitrite is converted to nitrate with the help of nitrite oxidizing bacteria like Nitrobacter. Thus, within 2 steps, ammonia is converted to nitrite. The last step of our cycle is denitrification. Organisms like Achromobacter and Pseudomonas carry out this reaction. It is a reduction reaction whereby nitrate is reduced to nitric acid, nitrous acid and finally leaves nitrogen back to the atmosphere. The process of nitrification is oftenly enhanced when there is more organic content, when the temperature is about 25 to 50 degrees Celsius, at neutral or alkaline pH and at low oxygen supply. Thus, these were the basic 5 steps of the nitrogen cycle. In spite of this, oftenly several heterotrophic bacteria convert nitrates to nitrite and back to ammonia under anaerobic conditions, that is in water-lagged regions. This is the reverse reactions occurring in nitrogen cycle. In spite of this, nitrogen also returns the atmosphere through volcanic eruptions and by emission through industrial combustion and gasoline engines. This was the basic thing about nitrogen cycle and the reactions which was in my look. Let us see the same reactions in a different angle from my colleague Paul Ben.
Dear students, just now you had very good and descriptive understanding of nitrogen cycle. But my question is, why we are talking about nitrogen cycle? Why microorganisms carry out any of the steps of this cycle? What will happen if we cannot get any nitrogenous compound, for example, in our diet? We human being, we are talking about we human being. You know that cell needs nitrogen to build up its very important compounds like amino acids, proteins, DNA, RNA, certain nucleotides, coenzymes, vitamins, etc. So, if no nitrogen is there, then how these building blocks, how these important biomolecules will be generated in our cells or in our self? Then suppose we are not getting nitrogenous compound in our diet, then we know that in atmosphere there is nitrogen in the gaseous form. Can we not use it? The answer to this is no, because we don't know how to use it. Meaning is, it is not required to know it in a theoretical way. The proper answer is, we are not having the machinery in our cells to utilize this gaseous nitrogen. Gaseous nitrogen is having valency 0, and the nitrogen which we need or many other forms of life needs is in -3 form. Then only if the molecule or the nitrogen is in such a form that it is having its valency -3, then only it can be incorporated in these biomolecules. So we have to depend on atmospheric nitrogen fixation through lightening process, or we have to depend on such forms of life which just now madam told you about nitrogen fixers, which are symbiotic nitrogen fixers, non-symbiotic nitrogen fixers, and one more is cyanobacteria, which fixes nitrogen with the help of heterocyst. So we all are thankful to these organisms that brings down this gaseous form of nitrogen in usable form, and that is known as conversion of nitrogen to ammonia. And in ammonia, if we count the valency of nitrogen, it is -3. So now we can use it, or other forms of life can use it. This is known as nitrogen fixation reaction. This is known as nitrogen fixation reaction. Now nitrogen is fixed in the form of ammonia. Number of organisms are capable, or many types of life are capable of fixing this ammonia into amino acid. From amino acid it forms protein. So plant forms proteins and animals depends upon plants. So from plants nitrogenous compound comes into animals. When plants dies or animal dies, it comes into soil finally. So soil is having organic nitrogenous compound. Now that's all. Plant can do nothing, animal can do nothing. But there are forms, little forms of life which can feed on this nitrogenous compound, and they can degrade this organic nitrogenous compound, and this process it is known as ammonification. But I want to tell you why this is done. Because by degradation of this protein, it will form ammonia plus some other compound. Now for these little forms, ammonia is not important. It do not carry out the reaction to produce ammonia. it want to have that other compound which will enter in catabolic reaction, that is energy generating reaction. For that purpose it does this reaction, right? So this is ammonification step, because ammonia is for the organism a side product. Now it is NH3. This little organism can do nothing about that. These are, when we talk, it is chemoheterotrophic organism, and it can do nothing more than this. So from organic matter it is now once again in the NH3 form, inorganic nitrogen, right? And other forms of life can feed on this ammonia and can convert it into NO2, that is oxidized form. This is oxidation reaction, hydrogen removal, oxygen addition, it is oxidation reaction. From NO2 it is converted to NO3. In both the reaction, the organisms which are involved, they are chemolithotrophic group of organism, means those organisms that derive electrons for their electron transport chain from inorganic chemicals. So they are nitrifying bacteria, they utilize electron for etc from this. Now NO3 is ultimately produced. This chemolithotrophic organism cannot do now anything of this NO3. So one more life can take the advantage of this, and under anaerobic condition they utilize this NO3 for electron transport chain once again. Why? Because instead of oxygen they utilize NO3 as a final electron acceptor and convert NO3 to reduced form NO2, still reduced form NO, still reduced form N2O, and from N2O to N2, which is once again atmospheric nitrogen back, and this is how completes nitrogen cycle. So one more option is certain plants can also utilize NO3 as it is seen in the cycle as a nitrogen source, and the reduction process in which final electron acceptor is NO3 can also convert NO3 into ammonia by reduction. There are two options. It can generate N2 or it can generate ammonia. There are two options. So this is how the cycle is interwoven or interlinked, and if ammonia is regenerated it can be easily taken up by the soil organisms or plants. Now you will learn about phosphorus cycle from my colleague Ami Purohit.
Thank you for showing a different angle of the reactions. Coming to the next is phosphorus cycle. Phosphorus is an important element of ATP, DNA, RNA, and it also forms phospholipids in cell membranes. A human body contains about 0.7 kg of phosphorus. Phosphorus is basically occurring in nature as a part of phosphates. Unlike other cycles, microorganisms do not oxidize or reduce phosphorus. It is merely a conversion of inorganic phosphorus to organic form and back to inorganic form. Or you can say a conversion of insoluble phosphorus to soluble and back to that. The transformation occurs as altering the solubility of inorganic compounds, mineralization of the organic compounds, and immobilization process. Let us have an overview of the phosphorus cycle. Phosphorus, which is present in the form of phosphates in the soil, is solubilized by nitric acid and H2SO4. The solubilized phosphorus is uptaken by plants. Plants are eaten up by the animals. When they both die out, the dead organic compounds composes of the organic phosphorus in the soil. They again represent as a form of organic phosphates in the soil. Moreover, the organic phosphates undergoes soil erosion, get dissolves, and becomes a part of marine phosphates. This marine phosphates get sedimented in the form of reservoir phosphate rocks at the depth of the marine environment. These phosphates are oftenly used for mining and manufacturing of fertilizers and detergents. The fertilizers are used by plants, so the phosphorus again passes to the plants, and the detergents are used by human beings. This fertilizers and phosphorus again undergoes soil erosion, whereby the phosphorus again enters the marine environment. Let us see each step. Phosphorus basically is present in the form of insoluble phosphates. These insoluble phosphates are solubilized by acids like nitrous acid and nitric acid, by nitrifying bacteria, and sulfuric acid produced by sulfur oxidizing bacteria. Thus, the insoluble phosphates get converted into soluble phosphates. This is a reaction which shows conversion of insoluble phosphates to soluble phosphates. It is oftenly carried out by organisms like Faeobacterium and Bacillus. The next is mineralization of organic phosphorus, that is converting the organic phosphorus to the inorganic form, which is carried out by enzyme phosphatase either at low or high pH. It is occurring more rapid under thermophilic range, at neutral pH, and in soil which contains rich organic phosphorus. Enzyme phosphatase is oftenly secreted by organisms like Arthrobacter and Streptomyces. And the third form is immobilization, whereby the phosphorus is assimilated in the nucleic acid, phospholipids, and other protoplasmic substances. That is, the phosphorus is accumulated in non-utilizable forms of the elements. This was the general movement of the phosphorus from insoluble form to soluble form and then back to insoluble form. Coming to the next element is iron. Iron proteins are found in all living organisms. Ion cycle is just a conversion of ferrous ion to ferric ion in aerobic conditions and back to ferrous ion under anaerobic conditions. Let us have an overview of the ion cycle. The ferrous ion is converted to ferric ion under aerobic conditions, and the ferric ion is converted to ferrous ion back under anaerobic conditions. In spite of this, in between, magnetites are formed under aerobic and anaerobic conditions. Let us see the steps in detail. First is ion oxidation, that is conversion of ferrous ion to ferric ion, which normally is carried out by Thiobacillus organism at 2 to 4.5 pH. This is the reaction which shows conversion of ferrous ion to ferric ions. The second is ion reduction, which normally occurs under anaerobic conditions, whereby Fe plus 3 is converted to Fe plus 2 by organisms like Bacillus and Clostridium. In spite of this, during ferric reductions and oxidations, magnetite and siderite are formed as the end products. The electrons required for this formation are supplied by ethanol and propanol. The humus containing ion compounds are often attacked by pseudomicrobium metallogenium organisms. They form ferric hydroxides which gets deposited coating the bacteria or in the form of filaments. Thus, organisms like pseudomicrobium and metallogenium forms filamentous ion oxides or coats the bacteria. This was the movement of the ion from Fe plus 2 to plus 3 and back to plus 2. Let us see these reactions from Parulben.
Dear students, just now you learned about some conversions between ion oxidized form to ion reduced form. Here it is seen in your slide, that is, it is conversion between Fe plus 2 to Fe plus 3. When plus is more, it is oxidation reaction. Why this ion gets oxidized? It could be one reason. By this oxidation, the organism will derive electron to pass in electron transport chain. Now, this is the speciality of the organisms which are known as chemolithotrophic organism. Litho means they utilize electron from inorganic sources. So, electron is derived from this oxidation. It enters in ETC. The organism will generate energy by that, and these are known as ion oxidizers. The same reaction, that is oxidation reaction, is also utilized in order to get electron by photosynthetic organisms also. Now, photosynthetic organism also oxidizes Fe plus 2, derives electron, and this electron continues photosynthetic cycles. Here there will be no generation of oxygen because it only generates when H2O is the electron donor. So, this is not oxygenic photosynthesis, but this is anoxygenic photosynthesis. So, oxidation reaction of Fe plus 2 for anoxygenic photosynthesis is also a possibility. Next is reduction of Fe plus 3 to Fe plus 2. Now, when the organisms are such a class that they have to live in anaerobic condition, then they are evolved in such a way that their final electron acceptor is not oxygen but it is Fe plus 3. So it will take up Fe plus 3 will take up electron and it will convert it into Fe plus 2 reduced form. So this is anaerobic respiration, the reaction. The purpose is to derive energy by anaerobic respiration. Now you will learn about one more cycle once again from Ami Purohit.
Thank you. Our last element is sulfur cycle. Sulfur is a component of major proteins. A human body contains about 70 kg of sulfur. Sulfur cycle involves the conversion of elemental sulfur to sulfate in anaerobic conditions, to H2S under anaerobic conditions, and back to elemental sulfur. Let us have an overview of the sulfur cycle. The elemental sulfur is converted to thiosulfate, to sulfite, and to sulfate under aerobic conditions. This sulfate is assimilated in the form of proteins. These proteins undergo desulfuration under aerobic and anaerobic conditions both. Under aerobic conditions, it forms back sulfate, and under anaerobic conditions, it forms H2S. H2S again undergoes sulfide assimilation to form proteins. Moreover, the hydrogen sulfide reduces to form elemental sulfate. This is also a back reaction where elemental sulfate again is forming hydrogen sulfide. In spite of all this, sulfate is oftenly converted to H2S, that is known as disseminatory sulfate reduction. Chemolithotropic group of organism oftenly converts hydrogen sulfide to sulfate. So this is the general overview of the sulfur cycle. Let us see each step in detail. First of all, let us see how does the sulfur enter the atmosphere. It enters in the form of hydrogen sulfide during volcanic eruptions and decomposition, as sulfur dioxide during volcanic eruptions and fossils and fuel burning, and as sulfate from sea sprays. Sulfur dioxide in the atmosphere combines with oxygen to produce sulfur trioxide, which further reacts with water vapor molecule to form H2SO4. H2SO4 is carried on the water with the earth with the help of rain. This is known as acid rain, which is damage for the plants, but an important step for sulfur cycle. This is the general reaction which shows conversion of elemental sulfur to H2SO4 as shown by Brayley and Trudenger. It is carried out by Thiobacillus thioxidans. Sulfate is assimilated in the plants in the form of sulfur containing amino acids and then into proteins. Moreover, sulfate may be reduced to hydrogen sulfide by soil microorganisms like desulfatomaculum as shown in the reactions. The reduced H2S, that is hydrogen sulfide, oftenly causes further decomposition and reduces elemental sulfur with the help of photosynthetic group of organisms like Bigotoa and Thiothrix. Thus, the elemental sulfur is again back in the atmosphere. Winogradsky column. It is a lab technique used for study of or cultivating of sulfur metabolizing bacteria developed by Serge Winogradsky scientist. He used calcium sulfate, plant parts, mud and water, added them in a column and incubated the column at room temperature under sunlight. After a few days, he observed different color zones in the column. At the end of few days incubation, he came to a conclusion that after the incubation, sulfur metabolizing group of bacteria, that is the organisms of sulfur cycle, can be cultivated in laboratory. Thus, it is a laboratory technique for cultivation of organisms involved in sulfur cycle. What is the importance of sulfur cycle? Sulfate is an important source of sulfur for plants. H2SO4 balances soil pH. H2SO4 helps in phosphate solubilization. SO2 absorbs UV rays. And sulfur cycle offsets global warming by increasing the cloud cover and maintaining a cool temperature. Let us see a different angle of these reactions from my colleague.
Next slide. Friends, just now you learned about sulfur cycle, in which you have studied about two terminology, rather one terminology, that is dissimilatory reaction, dissimilatory reduction. What is assimilation and what is dissimilation? Assimilation means to gather, to synthesize, right? And dissimilation means to generate energy. If the reaction is occurring for energy production, you will define it as a dissimilated reaction. If it is for some incorporation or some anabolic reaction, synthetic reaction, it is assimilated reaction. Now say here it is a conversion of SO4 to H2S. Now this could happen in some organism which do not get oxygen as an ultimate electron acceptor for their electron transport chain. So this anaerobic organism utilizes, instead of oxygen, similar type of inorganic oxidized molecule is SO4. It converts by taking electron gradually to reduced form, that is H2S. Now when SO4 is utilized, it is known as sulfur respiration, and the reduced product it is H2S. One more reaction is seen here in the slide. It is the utilization of H2S to derive electrons. Electron can be derived from Fe, electron can be derived from H2O, or electron can be derived from H2S. If it is derived from H2O, it would be oxygenic photosynthesis because it would generate oxygen instead of here, as you see, instead of sulfur. So as here there is no oxygen generation, it is an oxygenic photosynthesis, right? So reaction for their own purpose. One more reaction you will be able to see in the next slide is previous slide. It is sulfur oxidation once again. In the first reaction, sulfur is oxidized by chemolithotropes. Same principle, by sulfur oxidation, electron is obtained, electron will go down into electron transport chain, and this is how various organisms carries out each steps of the cycles, different cycles. No organism is interested in keeping the cycles going in the nature. They are only interested in their own life. So we will conclude the session with human impacts on different cycles.
Lastly, I would like to say that industries, vehicles and deforestations have shown about 35% rise in the carbon input in last 200 years. Human activities emit about 1/3 of the nitrous oxide during agricultural sector and industrial combustion. Overuse of phosphate fertilizers causes eutrophication that disturbs the ecosystem, and mining coal and extracting petroleum from earth has doubled the rate of sulfur. So I would conclude by saying that the natural input of these elements in the atmosphere is very less as compared to the human input of the elements. Can we do anything for this? It's up to you. Thank you.
Thank you.